Hello everyone, welcome back. This is part 2 of my discussion on electrochemistry. This is Dr. Rhonda Kofelt. In this part, we're going to look at the different types of cells, and we are going to write and interpret cell notations. So electrochemistry is the study of redox reactions. Is that what we just looked at? I know for a long time in in part 1, and because they are redox reactions, they produce an electrical current. And so that's the whole point. You're generating electrons and ions, and they're able to carry current. The conversion between the chemical energy associated with the reaction and the electrical energy is called a electrochemical cell. And so this chemical energy converts to electrical energy so that it can be used to perform work. Spontaneous redox takes place in what we call a Voltaic cell. It's spontaneous and it produces energy. It's also known as a galvanic cell. Okay, and so it's by moving those electrons across a wire, you're going to be producing electricity. Non-spontaneous redox reactions occur in what we call an electrolytic cell, and we have to add electrical energy to the electrolytic cell. So the spontaneous redox, okay, and this is something, notice that we have a copper solution in here, and that's why it's a pretty blue color, and we have a zinc strip, okay, and then what happens is on the surface of the zinc we have a redox reaction occurring, and so we lose the zinc and we replace it with a copper atom. And by doing that, we actually plate the copper on the surface of the zinc. So the zinc solid is going to be converting the zinc ions, and the copper ions from the solution are plating out as a solid on the surface of the zinc. And that is a textbook galvanic cell. So the electrical current that we're producing is basically kind of like when we were doing wavelengths, it's kind of like frequency. It's the amount of electric charge that passes a certain point at a given time. So whether the electrons are flowing through the water or the aqueous solution or through a wire, they are moving, okay? And so the how fast they're moving basically is the electrical current. So they're moving from one substance to another. And so we have, because it's movement between two things, we have that ability to generate the electrical current. The spontaneous redox does not require any external energy, and so it is spontaneous, therefore its delta G is negative. We learned all about that in chapter 18, the chapter on energy and thermo and all that, how that good stuff worked, okay? So in the voltaic galvanic cells, we produce electrical current from these spontaneous redox reactions. To use that, though, we have to separate where the oxidation occurs and where the reduction occurs. And so I'm going to show you kind of how we do that in just a minute. The oxidation and reduction are kept separate in the half cells. And so we've already figured out how to calculate those half reactions in the redox. And so the half cells are where each of those things is occurring. In order to have a circuit produced, then the electrons are going to flow through a wire. And the ions flowing through the solution are going to use what we call a salt bridge. I'm going to show you what all this looks like in just a second. The flow of electrons has to have some type of conductive electrode to allow this transfer to occur. And it either occurs through an external circuit or through a metal or graphite electrode. Okay, the electrochemical cell requires exchange of ions between, and so since we're exchanging ions in addition to the electrons, okay, then we have what's called the salt bridge thing. And I'm describing everything first, and then I'm going to show you what it looks like. All right, you have an anode. If you have some idea about if an anode or a cathode is positive or negative, just throw it away. All right, what you need to remember is the anode is where oxidation occurs, anox. We know how to figure that out from the last section. Okay, anions are going to be attracted to it. And so therefore in an electrolytic cell, it's connected to the positive end because the positive is going to pull those negative electrons toward, or anions I should say, towards it. In an electrolytic cell, the anode loses mass. Okay? A cathode, as you might guess, is where a reduction occurs. The cations are going to be attracted to that. It's connected to the negative end only in an electrolytic, okay? And it's the one that gains mass, okay? The electrode is where the electroplating that we talk about is occurring when you have an electrolytic cell. And remember, this is the one that you add energy to. So, for me, pictures make more sense. Some of you like words, that's why I tell you in words as well as pictures. All right, so in order for me to form a circuit, I have to have two things moving. Okay, I have electrons moving from one place to the other, and I have to have ions moving from one place to another. Okay, so and usually you're going to see these written anode, cathode, left to right. Okay, so on the left hand side, and I'll show you why that's important in just a second. The anode is your zinc. And what did I tell you? On the anode, remember anox. On the anode side, that's where oxidation is occurring. Oxidation goes with oil, so oxidation is loss. So your electrons are being lost from the zinc, and they are being sent to the copper electrode. Okay, so the copper electrode, the cathode, is where the reduction is taking place. And just as we saw a minute ago, the zinc, and what's up with this nitrate and this potassium? Okay, those are the anion and the cation, which you typically have in the solution. Okay, an anion, cation. So in this particular solution, we had KNO3 in there. Okay, and then we had a soluble zinc in there. And so since it's oxidizing, it's producing 2 electrons, and then the copper is being reduced by accepting 2 electrons and forming solid copper. So we're going to lose weight on the copper electrode, and we're going to gain weight on the copper electrode. Okay, so as we're doing this, and remember, this is a voltaic cell. Okay, voltaic, not electrolytic. Voltaic cell means no added energy. I don't have it plugged in. Okay, you don't see anything saying that this is plugged in. This light bulb right here, that's just showing me that the circuit is active. So when these electrons are flowing, that's going to light that light bulb up because it's going to produce enough current to light the light bulb. And a lot of times we do that just so you can see. This gizmo right here, and it can be a clear tube, or it can be like a piece of material or string or yarn soaked with something, KNO3. All right, and that is what allows the anion and the cation to go back and forth, because you have to connect them ionically and then connect them voltaically so that your electrons flow. So the purpose of the salt bridge is to complete the circuit and maintain the balance of the charge, because you got a plus and a negative. Okay? So a lot of people get a little mixed up about the difference between voltage and current. Okay, so we already said that current is the number of electrons that flows through a system per second. And so we talk about current in terms of amps, okay, or 1 coulomb per second. All right, and that's typically what we see in this. 1 amp is 6.242 times 10 to the 18 electrons per second. So that's a lot of electrons moving, right, if you have a 1 amp. And the more amps you have, the more electrons are flowing. Okay, the more electrons are flowing per second. So this is like the speed or frequency. The electrode surface is what really gives you the ability to flow more electrons. Larger batteries produce larger currents, okay? So you've got larger electrodes in there, or more efficient electrodes. The voltage is the difference between the potential energy of the products and the reactants, okay? We also call that the potential difference. And if you're in my course, you're going to be doing you're going to look at that lab and you're going to say, this is a biology lab. Okay, well, we use this in order to measure voltage changes, potential energies across membranes and things like that. And so I've given you a lab where you're actually going to see what that looks like when you put teeny tiny little electrodes in there and measure voltage changes across membranes. It's just a really interesting, I try to include different types of chemistry in your labs so that you see different aspects. Okay, so the unit of voltage is the volt, so that's not hard, and a volt is 1 joule per coulomb. Okay, so 1 joule per coulomb, and the voltage, you've got to have enough voltage to drive the electrons through that external circuit if you're going to be able to scavenge any of the work from it, any of the actual energy that you can use from it. Okay, so the amount of force pushing those electrons through the wire is called the EMF, the electromotive force. And you may have heard this term. They have EMF weapons. Okay, EMF weapons are those that pulse out and can turn off your electronic gizmos. Okay, so EMF, you also have EMF in cells, like you're going to look at when you look at the squid nerve tissue. All right, so the cell potential, I said, difference between potential energies of the anode and the cathode. All right, anode and the cathode. And so when we're calculating this based on the actual physical cell we have, we call that the cell potential. And the cell potential, the difference between the two energies, depends on how easy your oxidizing agent is reduced at the cathode, and the reducing agent is going to be oxidized at the anode. And so a lot of times how different their charges are, maybe a little bit, if you want to think about it like that. The cell potential under standard conditions is called the standard EMF, or the E0 of the cell. So remember, our little 0 there means standard conditions, and the cell is the entire thing, right? And so you standard conditions are room temperature, 25 degrees C, 1 atmosphere for gases, and 1 molar concentrations for anything that's an aqueous solution. And the sum of all the cell potentials for the half reactions, anode plus cathode, is your EMF of the cell. And you have tables that have all of these in it. For every half reaction, you're going to find there is a table that has that in there. So don't feel like you're going to have to memorize those things. So when we are communicating the type of cell we have, or if I want you to make a certain type of cell, we have a shorthand. And remember, we're talking about voltaic cells. We're still not adding any electricity to anything, okay? We're making our own electricity. So you have whatever the electrode is and a bar showing that it's separate. Then the electrolyte that is with this electrode, they go together. And then the double bar stands for the salt bridge. Then the other solution on the other side of the bridge, and a bar, and then the other electrode. Okay? So you've got your anode over here and your cathode over here, separated by the salt bridge. So if I say the anode is on the left, and this is a convention, so we all write it the same way so everybody knows, then that means that oxidation is on the left, and the cathode is on the right. That means reduction, because I said don't worry about if something's positive or negative. You just worry about if it's an anode, it's oxidizing. Okay? All right, and then this tells you what I just said about what those mean. All right, so for example, if you have a zinc electrode, so it's solid, it's a piece of metal, and you have a zinc solution in there, like we had zinc nitrate, I think, in there. And then you have a salt bridge, of course, because it's a voltaic cell. And then you had, say we had copper nitrate, it could be copper chloride, it doesn't matter. Okay. And then you had a copper electrode, so it's a piece of copper metal. And so you're telling me that that is a standard condition way to write this. Okay, so you're telling me you're at room temperature, all of your solutions are at 1 molar, or if you have a gas, it's 1 atmosphere, and you have salt bridge there, okay, and you've separated the two, so you should get a flow. And one of the reasons we write the oxidized thing, okay, the anode over here, is because you're going to flow in it. If you've got it set up right, you're going to flow anode to cathode. Now, if you hooked it up backwards, okay, it would still flow, but it would just flow the other way. So we can keep it straight, right? Here is the one that we drew, and you do need to be able to label the different parts of this. Okay, so you need to look at one and be able to tell me which was anode, which was cathode, where oxidation and reduction are going on, the salt bridge, you know, the direction of the electron flow, that kind of thing. Okay, so make sure you can identify those. All right, and so here is like the one we drew before with your zinc and your cathode, showing me all of those things, and then here is the shorthand that goes along with that. So typically your anode is going to be made of a metal that's been oxidized, and the cathode is made of the same metal that's going to be reduced. Okay? But we can do what's called an inert electrode. And the inert electrode acts as the electrode, but it does not participate in the reaction. Okay? It just provides a surface to transfer electrons back and forth. So if you see that word inert, means that it's non-reactive. We used to call the noble gases the inert gases because we didn't think they did anything until we found out that we could make them do things. So now that's why we call them noble gases instead of inert. But inert means it's not going to be reacted. So an example of an inert electrode is platinum. Platinum is pretty much a metal that doesn't participate in hardly anything. And it's very expensive because it's rare. It's a rare metal. But we use it in things where we don't want any corrosion or we don't want anything to happen. Okay? And so the half reaction with the manganese oxidation is going to occur as you see here. Okay. All right. from permanganate to manganese, okay? And then the iron is going to be what is producing. So the iron that's in solution is going to be what's producing the electrons, but it just goes to the surface. The electrons go to the surface of your inert electrode so that you have a source of electrons, but it's not going to decrease the mass of that electrode because it's just using the surface as something to kick those electrons to. Okay? Hopefully that makes sense. All right? But it doesn't, the platinum itself does not react with anything. All right? Okay. And so to write this one with an inert electrode, we have the iron one that's your typical with your iron solid and your iron aqueous solution. Then you have your salt bridge, okay? But on that other side, you have the whole thing going on. You have the permanganate making the Mn2 plus, right? And then you have your platinum electrode there. And so your platinum electrode is, oh sorry, I got it backwards, sorry. Your iron is going to an iron electrode, producing electrons. When the electrons get here, they'll kick off, okay, to take your Mn2 plus to MnO4 minus. But you're not plating anything. So, because you're not plating on this platinum because it's inert, right? So your electrons go from your iron solution to your iron electrode, and then those electrons are going to travel over to the platinum electrode, which is going to receive the electrons and then immediately eject those out, and then the whole reaction is going to take place right there in that solution. And so what you're doing is you're converting it from permanganate to manganese ion. And that's useful because if you have permanganate and you need manganese chloride salt or something like that for whatever, you can convert it all to the Mn2 plus and then add it to, then you can take that Mn2 plus and add it to like hydrochloric acid and make MnCl2. So it does have uses. It does have industrial uses to do this, and when they do it, they're using these inert electrodes because they're not interested in doing anything with the platinum. Plus, they're really expensive, so you don't want to use them up. And that's the end of part 2. Thanks for hanging with me.